Now, the lysosome is pretty powerful in terms of its ability to grade things, and that's the concept associated with the next topic, which involves the destruction of, in some cases, very large portions of the cytoplasm of a cell, basically on purpose. So I'm talking now about a mitochondria, peroxisome, maybe multiple mitochondria, the Golgi, ER if it gets in the way, that sort of thing. That phenomenon is referred to as autophagy. And the diagram that's over here on the right is taken from the Nobel lecture of the recipients of a Nobel Prize fairly recently, maybe in the last 2 or 3 years, based on their exploration, discovery, and understanding of autophagy. And I thought that diagram was actually really nice. And it makes the point that I was trying to make earlier, is that this phenomenon doesn't begin with plasma membrane endocytosis. It actually begins on the ER, but it ends with the lysosome fusing with a new organelle that we'll define in just a second to produce this destructive capacity that's important in cell recycling and also in nutrition. That is, basically, if the cell is starving, then it eats its mitochondria for food, kind of thing. So the overall process in detail is shown, I think, in a really nice diagram from this recent article that includes the mechanism and some of the regulatory processes associated with it. So the overall scheme of things is that a membrane system essentially emerges from the ER really kind of mysteriously and grows to be able to engulf the cytoplasmic stuff that gets in its way, essentially, that gets in close by it, in this double membrane system that then fuses with the lysosome for the destruction. So that's shown in this part of the diagram, and what is actually described here is specifically macro autophagy, to distinguish it from really 2 other things that I'll talk about in just a second. But what's shown here is an important regulatory concept associated with autophagy, and that is the fact that the lysosome is going to be involved in the late stages of the process. And if you don't want to do it, then you want to inhibit its involvement. And one of the ways that happens is through its association, the lysosomes, with a very, very important regulatory protein called mTOR, complex called mTORC1. So the first thing that has to happen in the case of autophagy, and remember, this is a starvation response, the first thing that has to happen is the inhibition of mTOR, and that I have shown on another slide coming up. But once that happens, there is the mystery that involves the formation of this sort of elongated vesicular system that's referred to as a phagophore. Here's mitochondria, maybe some RNA or DNA and proteins, let's say, a ribosome here and there. And that phagophore extends and then fuses with itself to form a double membrane system that encloses the things that are eventually going to be destroyed, because the lysosome is going to fuse with this autophagosome. That fusion involves some of our old friends, snares. It involves a tethering reaction that requires specific tethering factors like LC3. So LC3 is a very useful marker of autophagy, and you'll see it pretty much in all the literature about autophagy. And one of our other old friends, RAB7, in this case, that is involved in the specificity of the fusion between the lysosome and the autophagosome. So this is essentially just a donor-target reaction, just a very specific one. So that process produces something that's referred to as an autolysosome. So the combination of the autophagosome and lysosome means that all of those degradative processes that are stored safely inside the lysosome get dumped on the interior of this structure, essentially the lumen of this structure, which, remember, includes mitochondria and nucleic acids and proteins. So that they all get destroyed in the interior of this. Now you can just call it a really, really big lysosome if you want to. But in fact, what happens in the process of going from this to a recycled, renewed lysosome is pretty interesting too. And that is a budding reaction that involves two of our other old friends, phosphoinositol, in this case PIP2 with the 4,5 flavor, clathrin as the coat. So you get a clathrin-coated pit and vesicle that pinches off, as shown here. There's diamine, which I didn't talk about, doing the scission reaction, the pinching off. But once again, the specific involvement of PI45P2, but now in association with something that I wanted to talk about briefly, and that is identified as KIF5B on this diagram. So what KIF5B is a member of the family of proteins that are referred to as kinesins that I mentioned earlier as being one of the motor systems associated with vesicular trafficking and serving as the link between the vesicle, the lipid part usually, but also sometimes Rab, and the microtubules on the other side, and forming that that motorized system. So I wanted to take a little aside here just because of something that I wanted you to be aware of if you're not aware of it already, and bring up another sort of side topic. And that has to do with a special that is on PBS right now, a Ken Burns special called Gene. It's a two-part sequence. The first one has already been, the second one is going to be tonight, but it's all online, so you can watch it whenever you want. But one of the stars of that show, which is really good, is a gene and a person, a patient, that if you've seen it already, you'll recognize Susanna right away. But she is, I guess you could say, the unfortunate carrier of a gene that is KIP1A. So it's another mutation in a different kinesin, but the same function, that is the motor function. It just so happens that, not KIP, but KIF. KIF1A is present in the brain and spinal column and is involved in anterograde vesicular trafficking, which, if you know your motor neurons in the spinal column, one of them is very long and basically goes almost all the way down your leg. So if you don't have anterograde vesicular trafficking, you don't have neurotransmitters released at the end of that very, very long nerve, and there are serious degenerative problems associated with it. I won't say any more about that, but watch the show because it's great in all sorts of ways, but one of them involves some cell physiology. Okay, so back to our business. So we've been basically destroying the cytoplasm, a substantial part of the cytoplasm for nutritional purposes. We end up with those nutrients released into the cytoplasm and reconstituted lysosomes that basically just go back here so that the process, if it needs to, can start all over again. But there's an additional regulatory aspect to it that I wanted to finish this video with, and that's shown on this diagram. And it ties in autophagy with some other phenomena, not just starvation. So here's dietary restriction, which experimentally is what we mean by starvation, starving a mouse and seeing that they live much longer, for example. Humans seem to live longer when they're starved as well, although they don't live happily, if you ask me. But in any case, that restriction is associated with the inhibition of mTOR that I talked about was required for the purposes of releasing the lysosome so it can fuse right here to produce the autolysosome. But mTOR also inhibits other aspects of the initiation of autophagy, so inhibiting it stimulates autophagy in a number of ways. Exercise has been shown to do something similar, so one of the benefits of exercise is the ability to recycle nutrients, and that's associated with weight loss and overall good health. That process may or may not be related to this arrow, this part of the process, in which pretty important protein kinase called AMP kinase has also been shown to inhibit mTORC and therefore stimulate autophagy. We might come back to AMPK later on. And I don't think we're talking about metformin, but you might have heard about it on your own. It's a very important anti-diabetic drug. It's kind of a wonder drug when it comes right down to it. And one of the wonders of it is that it seems to simulate exercise, so that it's kind of like the exercise pill that keeps you from having to get on your treadmill. But it is anti-diabetic partly through this interaction right here. And then finally, there are things that activate mTOR for good reason. So insulin, growth factors, things that we would associate either with the absence of starvation, sort of glucose excess, or with the need to do something other than recycle our components. Growth factors would stimulate proliferation, so you don't want to do autophagy under those conditions. You don't want to release the chromosomes and then eat them up, for example, and that would be horrible. And then the product you can think of autophagy is amino acids from the degradation of all those proteins that have been engulfed. So those feed back to inhibit the autophagy by activating an inhibitor of autophagy in the form of mTOR-C. Okay, we'll stop there.